Okay, hello everyone. Oh, beautiful, beautiful audience. So what's gonna happen today, I should be standing in the camera, is that if you can hear me out in the museum, we're over at Science Today. We're gonna be hearing 6 talks from the Academy researchers, Academy scientists, all Pokemon adjacent talks, while we will be playing new Pokemon Snap live, all of which will be streamed on our new Twitch channel. So we're actually being very ambitious today. We're doing a lot. After every talk, there will be a Q&A section. So if you're live here in the audience, be sure to remember any questions that come up. If you're on Twitch, be sure to put your questions in the chat, and we'll get to them if there is time. And if you're live in person, if there's still space, be sure to sign up to play Pokemon Snap. Let's see. So our first guest speaker up today is Dr. Lauren Esposito. She's the curator and Schlinger Chair of Arachnology here at the Academy, going to present to us her favorite scorpions. Yeah.
Well, how am I supposed to change these slides? Hi. So I'm going to talk about scorpions. That's what I study. It's what I do. These are in, I wouldn't say like any really particular order, but they're some of my favorites. I haven't seen them all in real life. I mean, I've seen them in dead life, in a jar in our collection, but not all of them in nature. Stay right on top of the mic. Okay. Okay, so up first. Maybe not. What do you think? You can just say next. There you go. Alright, so up first is the Arizona bark scorpion. It is the only scorpion of like real medical concern here in the US. It's found like in Arizona, in parts of New Mexico, a little part of California. And it's the only like lethal scorpion that we have here in the US. There's scorpions everywhere. Actually, like California is a scorpion biodiversity hotspot, which I'm going to talk about in just a second. But the first one is the one that you might have to worry about if you're a child under the age of 6 in like a really remote area where you can't reach medical care within 24 hours. Next. This is what it looks like. It's a tiny little scorpion. So you can imagine this mouse, it's like the size of a house mouse. It's a small mouse. And the scorpion's even tinier. And you can actually, if you want to see it in real life, you can just go downstairs into the aquarium, or we have both of these things on exhibit together. The southern grasshopper mouse and the Arizona bark scorpion. And one of the reasons that we think that the Arizona bark scorpion has super toxic venom for mammals like us is because this adorable little mouse it's like most fearsome predator. These mice eat scorpions. They come out at night looking for scorpions, looking for a snack. They're ferocious little beasts, even though it looks kind of cute. Next. And I brought in a video clip to show you what happens. These mice don't care. Like, they'll get stung in the face over and over again. But something really cool is happening under the surface, which is that this mouse has evolved an immunity to scorpion venom, so that when the scorpion stings the mouse, instead of causing pain, which it does initially, but just for a moment, it converts that pain-causing agent and the scorpion venom into something that causes it to not feel any pain. So it takes something that's painful and creates a painkiller. And then it obviously just like devours these poor little scorpions. Hopefully y'all feel sad for them now. But not only is this mouse like a scorpion eater, it's also territorial. Like, that one. And they have like territories at night, so they're kind of like tiny little wolves, and they prowl around in their territories. And when it's a full moon, and if you have really good ears, sometimes you can hear them howling at the moon over their territory. You can play on that video. I think it doesn't really get much cuter than that. It's like a very tiny mouse, but also like absolutely terrifying if you're a scorpion. And probably one of the main reasons in terms of evolution that these scorpions are harmful to humans. Okay, next slide. Number 2 on my list, and I mentioned earlier that California is a scorpion biodiversity hotspot. It's one of the places in the world that has more species per like square mile than anywhere else in the world. And as evidence of that, just last year, I was working with 2 high school kids from here in the Bay Area who discovered 2 new species of California scorpion. They're these 2. There we go. Periobactinus soda and Periobactinus conclusus. They discovered these 2 in the Central Valley. They live in these kind of salt flat systems. So if you go to like a place that looks like the most inhospitable place in California desert, it's a salt flat, you can find these scorpions living all around it. And actually, we have another new species that we discovered this year that will be published in a few months, I hope. Number 3 is the shield-tailed scorpion. I've never seen these ones. These are found in the Middle East. It's pretty crazy, and I think one of my favorite facts about these scorpions, and I'm going to show you the picture of it in just a second so you can see how crazy it looks. It kind of looks like it's straight out of a Pokemon game, but my favorite fact about it is that it was originally discovered and described by a woman in 1932, which, like, when it comes to arachnology, there weren't a lot of women around. There definitely weren't a lot of women discovering and describing new species in the 1930s. But this one, which has this crazy shield tail, which is why it's called the shield-tailed scorpion, is found in Middle Eastern deserts. Next. All right, the next one is another of my favorites because it's also like a super weird-looking scorpion. It's these blind scorpions that live in cenote caves. If you've ever been to like the Yucatan and you've gone like swimming in a cenote or snorkeling in a cenote, there's scorpions down there, not in the water. They're just in the cave systems. And these scorpions have been living in cave systems for so long that they've completely lost all of their eyes and their eyeless. Can we see that picture? So here you have it. So like normally the eyes would be like up on the head, which is kind of at the front by the claws. But in this case, there are no eyes left whatsoever, not even like the eye structures. They're completely blind, and they have these kind of elongated claws and legs that they use for feeling around and sensing in the completely dark cave environments. Next slide. The next one is another pretty cool one, another extreme scorpion. So I think like most often people are like scorpion, desert, like big black thing, tumbleweeds blowing around. Like that's like the first thing that comes to mind. But there's actually scorpions like in some of the highest mountain ranges in the world, and this is the highest elevation scorpion in the world. It's found up in the high Andes, and these are parts of the Andes like where if we were to go up there, first we would need a coat, but secondly we might need oxygen because the oxygen levels are so low that it's hard for humans to breathe. Can we see that picture? And so here it is. It lives like up in these kind of alpine, treeless mountain tops where they're just like plants can't even really survive, just these like small forbs and grasses. And there's these little tiny scorpions up there just eking out a living up in the highest mountain range in the world. The next one I think is like, it's a classic, so you can't really go without it, right? There's no top 10 scorpion list without the emperor scorpion. They're called the emperor scorpion because they are the king of scorpions. They're the most heavy-bodied scorpions in the world. They're also one of the first scorpions to be formally recognized as endangered. And the reason that they're endangered, can we see the picture of it? Is because they're so big and beautiful that people have over-harvested them for the pet trade. These scorpions can live like 25 years in the wild. They live in sort of mixed-use burrow systems where the mom lives in the burrow with her babies and like in different cavities and chambers. So they're like, I would say kind of cuddly as far as scorpions are concerned. But they get over-harvested, and because they live in these burrow systems with multi-stage families, they can just like grab all of them at once by digging out the burrow. They're formally recognized under the IUCN Red List, and so their trade is limited now. But these guys are from sort of central North Africa. The next one is
my favorite because it belongs to the newest family of scorpions. So like, if you think about a family of animals, we're talking about like the rank of like all dogs. It's like if we woke up tomorrow and we're like, oh, dogs, like all dogs, like wolves, dogs, like every canid in the world we had never seen before. And that's true for scorpions. And we actually discovered a new family of scorpions, like completely discovered it. It wasn't like we knew about it before and later on we went back and looked at it and we're like, oh, this is distinct. No, like we found it in the middle of the Uzbeki desert in Central Asia. And since we first discovered it in the middle of the Uzbeki desert in Central Asia, we've subsequently found it in caves in Southeast Asia where we think that it's been hanging out since like before the dinosaurs came around, just hiding out in these caves, trying to stay alive as the climate changed around it. Can we see a picture of that one? They're not very big. If you've ever seen a mealworm, like if you've had a gecko or something like that and you fed it a mealworm, that's a mealworm right there, so you can see just how tiny these little scorpions are and why they were so easy to overlook for so long. Next. The next one's one of my favorites. It's probably my favorite because I named it, so that makes it like a no-brainer, right? But the reason that I love it the most is actually not because I named it, it's because of its crazy anatomy. Can we see a picture of that one? These are called club-tail scorpions, and they're called that because the end of their tail is actually like wider than the base of their tail, and it kind of looks like a club when it's stretched out. But the thing that I love about them is all scorpions have these structures underneath their body, like right on their belly, these little comb-like things down in the bottom right picture. And those comb-like things are normally used for sensory. They're used for like smelling and tasting the ground. They kind of touch the ground with it as they walk along. But in these guys, it's evolved for a really special function. They make noise with it, and they basically like hiss. So sometimes they're called hissing scorpions. And they hiss in response to predators. So like if predators are attacking them, they make this kind of shh, 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 shh noise. And they do that by rubbing these ridge structures. They've evolved these ridges. It almost looks like kind of like a fingerprint. Like it's like these tiny little micro ridges, and they rub it against a granular field on their stomach. It's kind of like a stick and washboard kind of noise, but like on a microscopic scale. So it instead sounds just kind of like a shh. Like a maraca or something like that. Next. This one is one of my favorites because it's called ghost scorpions. Like I feel like that's like, if you're like trying to pick a new name for scorpions, you want to find like the most metal name possible. And the reason that people call these ghost scorpions is because they're really, really hard to find, and so they're like elusive like a ghost. And the reason that they're super hard to find is that most scorpions, like the way that we look for scorpions out in nature is we go out at night when scorpions are active. We use an ultraviolet light, which is a black light, and scorpions fluoresce like a really bright green color, which makes it pretty easy to find them at night. We only learned about that like in the 1970s, and so since the 1970s, the number of scorpion species that we've discovered on Earth has like gone up exponentially because now we're able to see them really easily. But the one group of scorpions that doesn't do this is the ghost scorpions. And so the only way that you can see them or find them is through really hard, laborious work, looking under logs, flipping over rocks in their natural environment, which is in Southeast Asia, in really super dense tropical jungles. And so can we see the picture of them? I think they're really cute. They're like these cute little kind of chubby scorpions. Like they don't really move around very much. They just got like, they kind of sit there when you flip the log over, and they look just like the log, so they're impossible to see. They don't run away, so you don't see any movement, and you just kind of see like a shadow of a ghost, and then you know you found one. Next. All right, I think this is my last one. This is my top 10. And I think like I couldn't leave a list of scorpions without including this one, the flat rock scorpion. They're just so cool looking. Can we see a picture of it? And the reason that they're so cool looking is because they're super flat. And the reason they're flat is that they've adapted to living in rock crevices in southern Africa. And so these scorpions live in rock massives, and they just find like the tiniest little crack, and they're like dorsoventrally compressed, which means that they look like flat, like a piece of paper, and they just kind of like scrape out the rock crevice with their tail and their claws a little bit until they can squeeze their way into the crevice, and then they just hang out in there and they never leave. They just stay in there and wait for things to walk by, and they grab it with their claws and drag it in the hole. And I think that's it. That's my scorpion list.
So we have some time for some Q&A, either with our Twitch audience or with our audience in person for Dr. Lauren Esposito. Here you go.
I love this so much, and there were like 5 scorpions I was horrified of. What's the one that you would think you see in like movies that are like demonized a lot?
Definitely the emperor scorpion. Like if you've seen a scorpion in a movie, it's been that one, and like I can almost guarantee that the environment you saw it in was not the environment that it lives in, because scorpions like basically match their environment. So if you see like a scorpion that's really dark colored, it's probably living in like a really dark tropical jungle. If you see one that's light colored, it's probably living in like a light, very sunny kind of sand dune or something like that. And so when you see a black scorpion in a movie walking across like a desert landscape, it's definitely not living in that desert.
Someone in our Twitch audience wants to know, what's the estimate on the number of current known scorpion species?
So far, we've discovered about 2,600 scorpion species, which is kind of crazy if you consider that 100 years ago, scientists had only documented 250 or so. So like in the last 100 years, we've gone up an entire order of magnitude. And I like to say that we're like actually in the midst of a scorpion renaissance. Like we have learned so much more about scorpions in the last 100 years than we knew in the like preceding thousands of years.
Free Flower Girl asks, are there any other animals that have evolved specifically to hunt scorpions?
There are other animals that have evolved specifically to hunt scorpions. Actually, there's like a whole diversity. First, there's bats. Bats are like a huge scorpion predator. And like for all of you who thought like bats just eat things that fly in the night, they don't. Actually, there's a huge diversity of bats that are what we call gleaming bats, which means that instead of like swooping things out of the air at night, what they do is they fly around listening for noises of walking, like on the ground. And when they hear something walking on the ground, they like swoop down, they land on the ground, they pick it up, they stash it in this little kind of like flap at their butt, and then they carry it away and eat it in like a night roost. They probably like rip its head off first, but then they do the rest. There's also snakes. There's a specialized lineage of snakes that just eat scorpions and like centipedes. There's a bunch of other mammal predators of scorpions that have evolved venom resistance, like mongoose and some lizards. There's also scorpions are like a really tasty snack, and they have really, really high biomass. So there are like a lot of them in the places where they occur. And so if you can take advantage of eating that nutrition, then it's a really great snack.
Stop, you're making me hungry. Okay, I think that's about all the questions that we have time for. We're trying to get through a bunch of lightning talks. So sorry.
I'm cut off.
Okay, so if you're new here in person or new on Twitch, what's going on? You just put that right here. If you're new in person or new on our Twitch audience, what we're doing is we're hearing 6 lightning talks from Academy scientists and researchers that are all Pokemon themed, accompanied by live streaming of new Pokemon Snap that's being played by people, live NightLife guests here at the Academy. So up next, I believe we will have a talk by Maricela Abarca, all about fossil Pokemon.
Hi, everyone! Oh, hello, my fellow Pokemon people. Are there any people that came, like, just for geology? Probably not, but I thought I would ask. Okay, that's okay. How do the slides work? Okay. So my name is Maricela, and I'm representing the geology department today. I'm going to try to raise this up. Okay. And if you didn't know, this is the public floor of the museum, but 2 floors down is the geology department where I live. And you're going to get to see some of representation from our other collections today, too. But my collection is focused on things that are very, very dead and things that have been very, very dead for a long time. And I'm so excited to do this tonight again, because I get to combine 2 things that I'm super nerdy about, which is fossils and geology with Pokemon. And I was an OG Pokemon baby, or any Generation 1, thank you. So these are a little old school. There's some new ones sprinkled in, but it's mostly older generation Pokemon. And the Pure City Museum is kind of like the analog to this, right? It's Cass in the Pokemon universe, and they had fossils on display like we have here, and I'm going to show you some of the examples that we have in the collections here that inspired some of the fossil Pokemon from the show and the game. And this might not work. Thank you, Eric. So the first one is Ammonite, and if you got a Helix fossil in the game and you revived it, you got a little Ammonite, and just total disclaimer, like, Pokemon lets us live our fantasy of reviving and using DNA from rocks, right? We can't do that in real life, but you get to live your little Jurassic Park fantasy through Pokemon. And this one is inspired by Ammonite. The next slide, okay. And it's inspired in its anatomy and physiology and its name, right? Ammonites are some of my favorite fossils. There's a big representation in California. They were alive at the same time as dinosaurs and went extinct at the same time as dinosaurs. And there are so many fossils in California from this time period because at this time, California was underwater, it was marine. So that's why we don't have a lot of dinosaur fossils in California. There's some, but there's not a lot, but there's a whole lot of these. Oh, sorry. And these are some examples from the collections here. They're collected from kind of northern California inland. And if you think of that area now, it's like very not underwater. It's very flat, terrestrial, and agricultural. And when you find fossils like these there, it kind of hints at the past and what the landscape was like back then. And it kind of affected why that area is so agriculturally productive now. So that's one of the things that geology can teach us about the past and maybe plan for the future a little bit. But here you can see the two different forms that ammonites had. It had a closed form, so ammonite is kind of inspired by this one on the left, or on the right. And on the left, you can see a different form where the shell would open up, and it has like little spikes, and those are thought to be anti-predatory. And next slide, thank you. So the one that looks like a bugle, like soft serve shape on the right is just another form of shells that the ammonites would have. But if you bit it, you'd probably hurt your teeth because they're really hard, but something did take a bite of the one on the left, and this is one of my favorite examples in paleontology of how we can use and apply fossils like this, because it shows things that we can't observe, right? Unless you get in a time machine and go back in time, you're not going to see something eating this animal. But from the fossil, you can tell that something definitely took a bite out of it, and it was probably a marine reptile. And we just know because of the way that the bite marks look, the way that they're spaced, and where they are, that it was made by a marine reptile. And next slide, please. It was probably made by something like a lapras, and I love lapras. In the game, you don't need to revive her. She just is. She is beautiful. She is mysterious. And she's also, to me, an example of a plesiosaur. It's my opinion. But they have long necks, and they were apex predators of their time in the Cretaceous as well. What else? Oh yeah, the Cretaceous ocean was very scary because it was them, other really scary animals like mosasaurs, ichthyosaurs, think really big bodies, really big teeth. Next slide. And this is an example of the really big teeth. This is a skull in the collections here downstairs. And it's from Colombia in South America. It's where a lot of plesiosaurs are found. And it's just one of the examples of a plesiosaur skull. Great, next slide. Moving on, we have another rock water type. And the last time I gave this talk, people got kind of mad at me because I said it was not based on the trilobite. I actually, next slide. I said it reminded me of a horseshoe crab. I think it's both. It's whatever you want it to be, but I basing it on a horseshoe crab. This is a shout out to my arachnid people that are going to give a talk later, because they're not actually related to crabs. They're more closely or They're arthropods, but they're more related to spiders. And horseshoe crabs are those animals that are known as living fossils because their body type and plan and anatomy has been working for them for literally hundreds of millions of years. And it turns out that a piece of their anatomy is also helpful for us because their blood is used to isolate a compound to test vaccines for sterility, which was actually really important during COVID. And it's not unlike humans to contribute to the extinction and decline of animals, and unfortunately it looks like that's what's happening with horseshoe crabs. So hopefully with the attention from the COVID research, they'll become better managed. All right, so next slide. And this is a little example of a horseshoe crab fossil from a really famous deposit in Germany that preserves soft-shelled organisms. And this is just a little example. This one's only a couple of centimeters big, but again, you can kind of see the same body plan that horseshoe crabs have today. Next slide. Next, Aerodactyl. Fun fact, Aerodactyl is the fastest fossil the fastest fossil rock type in the game, so it's a very powerful Pokemon. It's one of my favorite, and it's revived from the old amber. And Aerodactyl is based on, you guys know this. Someone shout it out, please, help. Yeah, Pterodactyl. Next slide. So Pterodactyl is a kind of pterosaur. Some had teeth and some didn't. Aerodactyl is probably an example of one that did have teeth because it has little teeth, which means that it was probably a European pterosaur. But Quetzalcoatlus was the largest flying animal ever, and it didn't have teeth. So it somehow sustained that giant body type without needing teeth, eating something that didn't require it to have teeth. And I brought like a little bone, limb bone for scale. Just to like show the magnitude of this animal, it is about 33 to 36 feet long, like its wingspan. So yeah, all without teeth. Next slide. And this is just a little example, also probably from that that deposit in Germany, because you can see like the really fine preservation, and they're known to have been found there. Next slide. This is Arkin, revived from a plume fossil in the games, and it's one that I really like too because they illustrated it with colored feathers. And we know from fossils that have been found with really well-preserved color structure in the feathers that we know is responsible for structural color in feathers now. Next slide. And it's based on Archaeopteryx, which is like the transitional fossil. Yeah. It's like, yeah, there's so many photos of Archaeopteryx on the internet. I didn't include one here because we also don't have a good example of one. But I wanted to throw it in for Arkin. All right, next slide. And this is Leap. This one, unfortunately, is one that they got wrong, in my opinion, because it's revived from the root fossil, and it's a rock grass type, so she has, like, plant vibes, but it's not a plant. It's not based on a plant at all. Next slide. It's actually based on a crinoid, and even the common name of crinoids hints at plants. In the name of the sea lily, but those things are echinoderms. They're marine animals that are more related to sea stars and sea urchins than plants. So it's kind of one that I think they got wrong, but still cool. I'll give it to them. Next slide. And this is an example of one. See, so you can see why somebody might compare this to a plant. It has like a long stalk like a plant, and the top looks like flowers or leaves, but it's actually feeding appendages. Since they're marine, they kind of wave those around to get food out of the water column. And next slide. And this is another shout out to the the arthropod people that you're going to hear from later. But this is Anna. Next slide. Or next slide, yeah. And is based on Anomalocaris, and without this animal, we would not have T-Rex. Anomalocaris is actually an example of the apex predator on Earth 500 million years ago, and it's one of the kind of hard parts of paleontology. We'll never know if it wore its little mittens on the front appendages or on the side appendages. It's just something that we'll never be able to go back in time and observe, but we can have some fun and take some guesses. But yeah, next slide. It wasn't very big, right? This is only a couple of centimeters long, and if you look really close, you can see those appendages and its legs, and how this body form kind of led to being like an apex predator in the ocean. This is from the Burgess Shale in Canada. It's also a really famous deposit of Cambrian life forms. Next slide. And Cranidos is the last one that I have for you. I don't like including dinosaurs in my talks because I feel like they're overhyped. Everybody knows everything about dinosaurs. But I had to include this one because, next slide, if I saw this, I would think it's fake. Like, there's no way that this is real. There's no way that a dinosaur had a head like this. But we do know that dinosaurs had like heads like this because, next slide, we have the skull fossils of it. And so this kind of dinosaur is called a Pachycephalosaurus. And the jury's still out on what that dome is for. It's either for fighting or it's for attracting mates. But we're not really sure. And next slide, pretty sure that's it. Yeah, this is by no means an exhaustive list. It's just what I had time for. And honorable mention, mammal swine, because I do like the Ice Age mammals. But yeah, that's all I have for you. Is there time for questions? So much time for questions. I'm scared. Okay.
Again, anyone in the audience here in person, feel free to ask questions, or in our Twitch audience at home, let's start up here.
So as a geologist, I understand that you're studying these fossils as mineral formations in the ground, but how often do you have to switch over to a biologist's mindset and look at these fossils as instead animal byproducts?
That is a really good question because by training, I'm actually a biologist. So that's, so my background is in evolutionary biology, and I've kind of had to pick up the geology on the side. So I am actually more interested in like biological interactions that you can learn from fossils. Did I answer your question? Was there anything specific? That was good. Okay.
Someone in our Twitch audience brings up that in Sword and Shield, there were Pokemon like Dracovish, which were made incorrectly, like assembled wrong.
Yeah.
Like where does that come from?
Yeah, okay, that's why I didn't include them too, because I hate them. I think they're really awkward. But yeah, I think it's just like a weird amalgamation of like trying to include fossil stuff. I don't know if they're assembled wrong, but they are weird. Like they look weird, their types are weird. It's just weird. It's a mess. Yeah.
Someone else in our Twitch audience asks, how do plesiosaurs hunt? Did they chase their prey, or do we know anything about them?
Plesiosaurs? It's been kind of debated on how they hunt. There's like one hypothesis that said that their whole body was on the seafloor and their necks were so long that they would like fish with their necks. But then like there's also support for the other way around where their bodies are in the water column and their necks were like digging in the ocean floor. So it's just one of those things about paleontology. You see these fossils and you can kind of guess, but we might never really know. Yeah.
You're getting a lot of compliments in our audience for your Psyduck sweatshirt, by the way.
You. I had to represent Psyduck, even though he's not based on a fossil. I love him.
We have any other questions? No? If not, thank you so much, Maricela.
Thank you all.
Okay. Great. Our next talk will start at 7:40. It'll be Emma Roman talking about PokéSymbionts. Okay, hello everyone in person at the Academy or online at Twitch. Thank you so much for joining us. What's happening right now is we're hearing lightning talks from Academy scientists and researchers that are all Pokemon adjacent, and we're also, for people live at the Academy, we're playing new Pokemon Snap, all of which is being streamed on our Twitch channel. So thank you all for joining us. We have a talk, we're about to hear a talk from Emma Roman all about symbiosis and how that connects to Pokemon. So take it away. We'll also have questions afterwards, so remember them if you think of any.
Hello, hi everybody. So as we just said, I'm going to talk about symbiosis today, and I actually have a surprise at the end for both people on Twitch and people here. Quick spoiler, I have glowing bacteria here, so stay to the end for that. Yeah, let's go ahead and get started. So next slide, please. Let's see. Also, okay, so by show of hands, who's heard of symbiosis? Just the word before. Yeah, awesome. Okay, cool. So the definition of symbiosis, for those who don't know, and also just so we're all on the same page, is it's a biological interaction when two different organisms live closely together. So what that basically means is two different things living together, right? Next slide, please. So there's three types of symbiosis. If you're like me, I tend to think of this mutualism one, which is the double plus signs, the two little green animal things. And this is when two different organisms live together for the mutual benefit of each other. So here they're both green, they're both happy. There's also commensalism, which is depicted here by this like gray long-legged animal creature. And that's because in this type of symbiosis, one organism benefits, which is the little green man at the bottom, and then the other one just couldn't be bothered. No positive, no negative, completely no. And then the last one is parasitism, which is sometimes we don't think about this when it comes to symbiosis because it's one positive and one negative. But we'll get into some examples of that soon. So next slide, please. Okay, so just, you don't have to raise your hand, but just go ahead and think. I'm going to show some examples here on the slide of symbiosis that we might know of in the world around us. So here we have an anemone and a clownfish. So go ahead to the next slide, please. Here we have an alligator and a bird, and the next slide, and here we have a lichen, if people know what that one is. And the next slide, please. So you might know this because you've seen them on TV. You've seen them on Netflix, on like a BBC documentary. Maybe David Attenborough has talked about this before. And then go ahead to the next slide, please. What's really cool, though, is we've seen these on TV. We know this exists in the real world, and we actually use this as inspiration for entertainment. Next slide, please. So here's some examples I could think of when we use biology, ecology, symbiosis in pop culture. Next slide, please. Today, talking about Pokemon, as I'm sure you all are aware of. Let's go to the next slide. So first, let's talk about mutualisms. That's a double plus sign, double green, everyone's happy, everyone's thriving. So in Pokemon, there is these two Pokemon, Fungus and Lurantis. And what happens here is the Fungus and Lurantis will create this connection via their root system. So the Lurantis is actually a grass-type Pokemon. And this actually happens in the real world. We use agriculture a lot as an example, where we have a fungus that will connect with the plant, and then they make each other happy. They exchange nutrients, food, water, all that kind of stuff. And that's what happens here in the world of Pokemon. And the Lurantis actually needs the fungus to reproduce. So it's pretty cool. Let's go to the next slide. So next we have commensalism. If we remember, that's that one benefits positively and one just couldn't be bothered. So the one that couldn't be bothered here is the Ivysaur. So what happens in this relationship is the Beautifly, when it's a pupa, when it's a baby, which I believe is called a Wurmple. We'll see on the next slide. But when the Wurmple is a little, it eats the leaves of the Ivysaur. So the Ivysaur will drop its leaves. And what's really crazy is the Ivysaur leaves have this chemical compound that's like a poison, it's like a toxin. And so the Beautifly will eat those leaves, and it doesn't hurt the baby Beautifly, where it will actually hurt other insects in the world of Pokemon. So what's cool is then when I believe Wurmple becomes a Beautifly, it actually contains some of this toxin inside of it and doesn't taste very well to other animals that want to try and eat it. This is something we do see in the real world, where we have butterflies or other insects which just don't taste very great. And as a predator, you don't want to eat something that tastes super bitter, right? Let's go to the next slide. So what's cool is I really got deep into the Pokédex for this one, but the Wurmple actually has two evolutions. It can become that Beautifly I was talking about, right? That tastes bad because it eats that poison from those leaves, right? But I don't really understand why, but sometimes it becomes a Dustox instead. And what's cool is the Dustox is actually a poison type. So I'm not exactly sure if it's a poison type because it eats the leaves when it's a baby, but that's pretty cool to think about. So we can go to the next slide. So next we're going to talk about parasitism. So here is, I think, a very classic example of parasitism with the parasect. So what happens here is there's an insect. This is like a crab-looking insect, and it is completely overtaken by this mushroom, which is living on its back, and it completely mind controls it. By raising hands, has anyone seen The Last of Us? This is kind of the same thing that happens in The Last of Us. And it's based off of this fungus that exists in the real world and takes over ants, actually. And so in this relationship, as we can see by the cute little picture, the red guy is not very happy about the green guy eating it. The red guy would be the insect here, and the green little man would be the fungus here. So the fungus is benefiting completely at the cost of the insect, which is still its type of symbiosis. Let's go to the next slide. And then just because I'm extra, I wanted to show another example of parasitism because it very loosely ties into the work that I do here at Cal Academy. So here is the Morlo and the Alolan Executor. I don't know, who knows how to pronounce this word? Executor? I think it's Executor. Executor, I haven't played Pokemon in a long time. So in this relationship, remember in the beginning how I was talking about Fungus and Lorantis and how they'll share nutrients and water and everyone's happy and grows big and strong together? Well, in this relationship, the Morolo will take from the plant and not let the plant take from it. And in fact, when the plant tries to take resources from the fungus, it actually makes the plant sick. And so that's a type of parasitism where it's just leaching nutrients from the Alolan Executor. And so I wanted to include this example because the Morolo is a bioluminescent fungus, which means that it actually glows in the dark. And we can go to the next slide, actually. So what I study here at Cal Academy is this mutualism. So remember, double plus signs, everyone's good, everyone's happy. I study that between this itty little fish, Scyphemia tubifer, and this bacteria, Photobacterium mandapenensis, which is bioluminescent. So unlike the Morolo, my bacteria is positive, good kind of stuff for this fish. And so, the what? It is a little bit of favoritism, but what can I say? Bioluminescence is really cool. So, whoops. And on that note, I actually have, we can go to the next slide. I actually have bacteria with me that glows in the dark. And so I had some fun, and I decided to see if I could make some Pokemon, because my system doesn't exist in the world of Pokemon yet. And so I decided I'd make it into Pokemon myself, so I'm going to pass these around. And so what's in these boxes is what you see on the screen. They are glowing, but your eyes are not necessarily adjusted to them yet, so it might be really hard to see. But this is exactly what you would be seeing here. And so the blue, the Squirtle, is completely natural. That 100% happens in nature, whereas the green, the Bulbasaur, we've actually in the lab added some protein that you don't need to know. Basically, we made it green in the lab, which is really cool. So we can go into the next slide. So that's all I have for y'all. It was really quick, but also here's my social if you want. I'm going to post these pictures online if you want to brag to your friends, feel free. And then I got everything from Bulbapedia and Pokédex. So I'm by no means an expert on this, but I do love symbiosis, I love biology, and I love entertainment and pop culture. So thank you.
Thank you so much, Emma. We have plenty of time for questions. So if anyone in our live studio audience has questions, or if anyone on our Twitch stream at home has questions.
In your research, how does the bacteria and the fish, how do they benefit each other? I'm so glad you asked.
I'll keep this pretty short. So basically what's happening is I want you to imagine sort of where your belly button is, that you have like an extra organ, like an extra stomach almost, that's completely empty. And so what happens is the fish has this organ that it will put that bacteria in, and when the fish is swimming on the
Out of the ocean, what happens is it eats at nighttime.
and so there's a shadow on the ground. So it'll use its stomach as a light source to cancel out its shadow to make it kind of invisible to predators. It's what's happening there. I know, right? Right? And what's cool too is like it's a one bacteria to one fish system, right? But it's related to your stomach. And so once we understand how that works, we can begin to understand how your stomach works. Because inside of your stomach right now is, gosh, like hundreds of thousands of bacteria that are helping you digest your food, get all of your nutrients, super like mutualistic, double plus signs there kind of deal. So, yeah.
You said hundreds of thousands of bacteria. Do you mean hundreds of thousands of species of bacteria or hundreds of thousands of individual?
Individual bacteria, yeah. I don't know the exact number, but there are studies where they've said there are certain families of bacteria that commonly occur in your gut, and you can actually look at the percentages. And I can't speak to this like super well, but there are studies about what is in your gut and your mental health, and how there's a connection between your gut microbiome and actually your mental health as well, which is super cool.
Someone in our Twitch audience asks a question. You might be a little biased, but try to be as objective as
I'll try.
possible. What do you think is the most interesting parasitic relationship in real life?
Most interesting parasitic relationship in real life. I don't know. I guess I think leeches are pretty cool. Just the idea of how leeches attach to large organisms, and they actually have this like jaw that is like 3 pieces,
which is really freaky, and they'll like latch on. That's why if you get a leech on you, you shouldn't like rip it off, kind of deal. So maybe also that's my favorite, but also like if I'm being biased, the Last of Us is pretty popular right now, and so that's a pretty fun parasite to think about.
Is that based on any real world organism?
Which one?
The fungus from The Last of Us?
Oh yeah, absolutely. So there's these ants that actually will get infected with cordyceps, just like in the show. And what happens, I know, don't worry, we haven't found any in humans, so don't worry. And what happens is the fungus grows inside of the ant crawls up a tree, I believe, and then it'll die there, and then it'll have like, the fungus will sprout out of its head and release its spores to go to other ants in the rainforest.
Is there like a connection though between them where they're like
Between the ants or between the fungus?
the fungus that's infected the ants, like so various ants that have been infected by the
Oh, like you asking if there's like a hive mind kind of thing going on?
such as in the show.
Like I actually don't know, but I wouldn't be surprised. That's my favorite thing about biology is I want to say no, but then every time you say no, there's always an exception to the rule. So I don't actually know. I'd have to look, but I don't think quite like the show where they're like going under people's like fingernails and stuff like that. Yeah.
You really engaged the Last of Us fandom on Twitch tonight.
Oh, I'm so ready. I love The Last of Us.
A lot of more just like observation, like wow, it's so cool, it's so gross.
Oh,
so weird.
thank you. Don't worry, I say ew at work all the time. I'll like grow the bacteria that are green, that are like biologically, like we've changed them in the lab, and the liquid is like bright green. And one of my friends came up and said, that looks like Gatorade. And now I'm like, oh, I can't drink Gatorade anymore.
Can you speak a little bit more about the mechanism that the bacteria uses to bioluminesce?
Yes, so there's actually two different ways things glow in nature. There's intrinsically, where it will have this enzyme called a luciferase. Basically, all you need to know is it's this thing inside of the organism that eats this certain type of chemical and creates light. So you can do that either just by yourself, or you can do that with a symbiont, with a bacteria. And so the bacteria I work with has that little enzyme, that thing that will eat the chemical and make it glow. And we actually have downstairs in the bioluminescent exhibit, we have some flashlight fish, which actually have the same bacteria that all of you just got to look at, same family, where it lives in their under eye socket, and they'll open and close it. So after this, if you want to go look at some in a fish, they're downstairs, super cool. But yeah, basically, there's a chemical reaction there that happens, and then depending on the chemical will change the color actually. So mine are blue, there's green and I think yellow, which happen more on like terrestrial settings, and I think red sometimes in the deep, deep ocean. Mm-hmm.
That is fascinating. Thank you so much, Emma, for sharing.
Thank you.
Okay, our next talk will be at 8 p.m. Jacob Gorneau is going to talk about a Pokédex worth of spiders. Thank you, Emma. Going on here is we're having 20-minute lightning talks, all about all from Academy researchers and scientists that are like Pokemon adjacent, and the people who are in person here in the Academy have the opportunity to play new Pokemon Snap, and we're streaming both of those at the same time. We're doing a lot. Up next, right now, we have another talk all about spiders and the spiders in Pokemon from Jacob Gorneau.
Alrighty, thank you, Darian. Let me know if I'm too far away from the mic or anything, but I am a grad student in Lauren Esposito's lab, so if you were at the first Scorpion talk, that's who I work with, and we'll also have another grad student, Kate, coming up after me. And so I'm really excited to be talking to you about a real-world Pokédex today. So next slide. And so there are a lot of different ways that we catalog biodiversity on Earth, and a lot of it centers around learning how we know what we don't know. And part of that is that there's a majority of biodiversity that's in the global south. And you can see that on this map right here of iNaturalist observations. Although we've got a pretty good spread across the world, the most dense observations are those most orange, reddish, and they're in North America and Europe. And we know that the greatest areas of biodiversity are in South America, tropical Africa, and tropical Asia. And so we've kind of just through stuff like iNaturalist and this biodiversity information facility, GBIF, technology has really advanced the accessibility and the availability of this information. Next slide. And so I just wanted to, I'm sure many of you or all of you are familiar with Pokemon and Pokédexes, but basically here are some important elements of a Pokédex. Whenever you first catch a Pokemon, the Pokemon is recorded there, and it's organized in a standardized manner that allows you to organize by type. It also provides stats and different types of information for each of the Pokemon that you have caught. And at its most basic level, it gives you an idea of what you've caught and what is left for you to catch. And so you have a really good idea of where you stand in the biodiversity of Pokemon. Next slide. And so in the real world, we actually do have Pokédexes, except they're given the less exciting name of biodiversity catalogs. And essentially, these allow for the documentation of taxonomic changes through time. So they are able to document when someone describes a new species. They document other changes that happen. So sometimes when someone describes a new species, we find out later that it's actually more closely related to other species. And so we'll move it into a different group. And so biodiversity catalogs are able to record this information. And they also standardize this information and make it accessible for researchers so that everyone's working off of the same rule book. And oftentimes these catalogs were they weren't online. They were actually in libraries and really difficult for people to access. But they've recently become online, and that allows a lot more people to be able to use them, and it allows fields of taxonomy to go much faster because people can work off of the same document. Next. And so the World Spider Catalog is the biodiversity catalog I'll be talking with you today about. And this is actually the largest taxonomic database. So this is the largest real-world Pokédex that is out there right now for living animals. There are over 50,000 species of spider, and every single species of spider has a record in this database. And it actually was pretty early online. It first became online in 2000, and since 2014, it's also provided access to over 16,000 scientific articles. And as long as you have like a free membership with this community, you're able to access all of these articles. And some of these articles, otherwise you would have to pay for a journal subscription or work at a university to have access to. So it's really changing who can be doing this science and who can be studying spiders. Next. So here's a little snapshot. It's a little less exciting than the Pokédex interface you might be familiar with, but essentially it gives us the most important information. The family where the spider is in, the genus as well as the species name. It provides all of the references, so every single time a taxonomist has
dealt with that spider, it's recorded here, and it also provides a distribution string, so it gives you an idea of where you can find. And so for this species, it has Europe, Turkey, Russia, Iran, Central Asia, and Korea. And so it gives you a really good idea and a bunch of quick little stats about the organism that you're studying. Next slide. And so kind of what I've been getting at is that unlike Pokédexes that are available to every single player and are accessible to every single player, these biodiversity catalogs are not. And so that's been something that a lot of scientists are trying to break down right now, because it creates this dynamic where a lot of the biodiversity is in the global south, but these resources and these biodiversity catalogs are in the global north. And so people in those communities cannot access them and cannot study the biodiversity in their area. And so developing these biodiversity catalogs for groups like spiders and making them accessible online really transforms that and allows people in their local communities to do taxonomy and describe new species. Next. And so, kind of to that end, why should we know how many spiders exist? Part of that is to get an idea of how much is left to be described, and also a lot of scientists like to call things understudied. But of course, if you want to study something more, you're going to call it understudied because you want people to give you money to learn more about it. And so it's usually qualitative. And so what we wanted to do with our research is develop like a more quantifiable metric that could be used for people to identify what spiders are understudied versus what spiders are not. And it's just like in Pokemon, where it's helpful to figure out how much is left to discover and where, so you can really focus your efforts. Because spider taxonomists, as you might imagine, aren't given too much money to do their work with, and so they have to be really frugal and conscientious about where they're spending their money. Next. And so the goals for our project were to create this metric of taxonomic effort for spider taxonomy. And essentially, in terms of effort, what you can think about is where, when, and what are scientists describing. And the idea is that if we have an idea of where this effort is being put, we can identify families that need additional study, particularly in the area of species description. We also wanted to investigate this by clade, or their evolutionary history, and also zoogeography, so different regions of the world. And our goal overall is just to inspire people to create these biodiversity catalogs for other groups, because it's actually pretty rare for a group of organisms to have a biodiversity catalog. Next. One back, please. Alrighty. So this is a bit of a convoluted slide, but I'll walk you through it. We're essentially proposing this ratio of valid species descriptions, so essentially the number of species in a family, to other taxonomic changes. So I mentioned sometimes people will move a species into one genus after realizing it's more closely related to those species. Basically, all of those changes don't have anything to do with like new species discovery, and so we basically created this ratio of counts that are related to new species discovery and other things that taxonomists do in their work. And by log transforming this metric, we have a positive value in pink that indicates that this group probably will result in more new species descriptions, so we should focus our species description efforts there. And green indicating that there are less species descriptions, so it might be time to start investigating the relationships of this group, because we already have a good idea of the number of species there. And so all in all, this helps us find places on the map that might have more species for us to discover. Next slide, please. And so I just described that metric, and what we have up here is a plot of every single spider family. And what I want you to focus on is the fact that in this like pinkish fuchsia color toward the top, those are all of the families that we've identified as needing much more investment in species discovery. And so we were able to develop this metric to have an idea of what groups need that work. And these other groups, it's still important to study them, but it likely means that we have a good idea of what species are in that group, and that we can start investigating other aspects of their biology, such as how they're related to each other and other things, because we've already sampled the number of species in their group already. Next. We also looked at this. This is essentially the same plot, but it's on a phylogeny, or the evolutionary history of these spiders. And so this allows us to look at if there are certain evolutionary groups of spiders that are studied more or groups that need more study. And what we did find was that there is some bias in this evolutionary context. Next. One back, please. So we found that this group called the Synspermiata, which includes daddy longleg spiders, they likely have they likely need more of an effort in species description, and there are likely more of these spiders to be discovered. And we can tell that because by looking into the tree, if you look kind of deeper, more toward the center of the circle, there's still these pinkish areas that indicate that positive value, that there are still more species relative to these other taxonomic things that people do. Next. We also observed this for tarantulas and their relatives, that a lot of tarantula relatives also probably have many more species left to be described. Next. And then kind of on the flip side, here's a group of spiders that seem to be well documented in the area of species discovery. So this is a wolf spider. There are wolf spiders and their relatives seem to be much more well documented relatively. And you can see that even in the phylogeny, pretty deep toward the center of the plot, you can still see that green color indicating that there are fewer species relative to the other taxonomic changes that scientists will make. Next. So we also looked at trends by region because we wanted to see if there were any areas of the map that needed more work. Next. And so kind of to start, what we did was we just plotted, it's called a species accumulation curve, but it's basically throughout the history of when people describe spiders, we cumulatively added every single species in each zoogeographic region. So Australian, Nearctic is primarily North American, temperate North America, etc. And what we can see is that in the center of this plot, there seems to be a plateau for a lot of regions that had been colonized in between the 16 and 1800s. And we think that this plateau, about 75 to 120 years ago, is pretty directly correlated with these European powers leaving these colonial regions, and these colonial regions now needing to set up their own spider taxonomy programs, their own resources that they were left without when the colonial powers left. And I also want to make a caution about generally making biological inferences from political designations. One of these is that zoogeographic boundaries are hypothesized to have biological implications, but oftentimes these are connected with marine trade routes and different things like that rather than explicitly biological hypotheses. And also, it's really difficult when you have a species that says, oh, it's been described from China. We don't know whether it's temperate China or tropical China. And so in a lot of those instances, we could not go through every single 50,000 spider species and do that. And so we ended up needing to count it multiple times. And so that's an area of future direction that we'd like to investigate. Next slide, please. So we also, this is that same plot that I showed you earlier, but with the share and percentage of species from the global north and the global south. And
Overall, what this shows is that
the data that we're working with are also biased. So we have overrepresentation of families in the global north that seem to warrant more effort in that area of species description. And so that's not what we were expecting. We were expecting more effort to be needed to be invested in the global south. But part of this is just, we think, because you can't disentangle the colonial histories of taxonomy and of the history of science from the work that we're doing. And so that's really what's going on here, we believe. Next slide. We were also able to use this ratio to estimate species diversity, and essentially we just used this ratio, multiplied it by the current number of species described, and then we were able to get a pretty solid idea of how many species are left to be described. And this might be good news to you if you're afraid of spiders. We did predict a total predicted spider diversity of around 85,000, which means that there's about 35,000 spiders left to be described, but it's significantly lower than another metric, 120,000, that was proposed by this group that basically just evenly multiplied the current number of species by basically 3. And so since we calibrated it specifically to each family, we feel more confident in our estimates. Next. And so overall, these biodiversity catalogs, they contain taxonomic as well as sometimes these other physiological, behavioral, and ecological types of information. And making these catalogs accessible allows many more people to be doing this type of science. And so these Pokédexes and making them virtual and accessible in the area of taxonomy is actually incredibly important to biodiversity science. And next slide. And so we don't have too many direct outputs of this research. It was just published a couple of weeks ago. And so we're excited to see what people have to say about it and how people use this work to inform their own work. But that's kind of why we don't have any direct updates just yet. And so stay tuned. And I think I have one more slide, but feel free to ask me any questions. I also have some social media handles. And thanks for listening.
Thank you so much, Jacob. If anyone in our in-person audience or our audience on Twitch has any questions, be sure to leave them in the chat or just raise your hand if you're in person. Covered a lot of ground.
We would like to know your favorite spider.
Okay, that is a really good and also a really tough question. I think that this is maybe a bit of a cop-out because I studied this group for my master's, but there's this group of spiders called the dictynid spiders that Kate will actually be talking about a little bit in this next talk. And there's a spider in that group called the diving bell spider, and it basically creates this air bubble that it puts over its breathing organs, which are actually on basically its stomach, and it is able to live most of its life underwater. And so it's this really, really interesting behavior that is not found in any other spiders, and so I would say that's one of my favorite spiders. Thank you.
Okay, Free Flower Girl asks, how important is community science to modern taxonomy?
That's a really good question. So community science is incredibly important to taxonomy, and it's actually been important ever since taxonomy began, especially in entomology and arachnology. A lot of the people who have described spiders and scorpions and insects over time have actually been hobbyists who just happen to have a really strong fascination with arthropods. And so throughout that history, people have been interested, but even today, we use sites like iNaturalist to plan our field work. We'll type whatever species we're interested in seeing, and we use that to determine where we put our resources and where we search for these scorpions. So much so that sometimes we can go right onto the iNaturalist app and be pretty confident that we found the same exact scorpion that someone saw even a year ago under the same exact log that it was based on the GPS. So it's actually super, super important.
And
No, it's okay. Do you have a favorite spider Pokemon?
I am a big fan of, I don't know too much about Pokemon, but I think it's a new Pokemon called Spidops, which is a huntsman spider, and I actually studied huntsman spiders in my undergrad, and so that one's been really special to me. I also like the color green, so they're green.
Awesome.
okay, we have time for one more question from our Twitch audience. A lot of people want to know if you're wearing the same shirt right now as you are in that picture.
I actually am not. This is much heavier, much more SF friendly. This is a very light field shirt, and I would be very cold if I was wearing that, but I get sunburned if I don't wear that in the field.
Okay, thank you so much, Jacob.
Of course.
In a few minutes, we'll hear from Kate Montana. Okay, hello for everyone joining us in person at the Academy, or if you're joining us newly online on Twitch. We're here streaming lightning talks that are all Pokemon themed from Academy researchers and scientists, while in person, some of our live NightLife guests are playing new Pokémon Snap, and we're streaming that too in between. But right now, we will hear from Kate Montana on how she catches them all. If you have any questions, be sure to put them in the chat or remember them so you can ask them in person. So let's give it up for Kate.
Thank you, Darian.
Awesome. Hello. Nice to see you all here in person and also online. Thank you for joining us. I am going to be talking about some stories from my time in the field as a field biologist and tell you a few ways that I've caught, maybe not all of them, but some animals. All right, next slide, please.
So, a little bit about me. I am an arachnologist in training, which means that I'm getting my master's degree here at the Cal Academy. I am doing my research downstairs in the basement, and I also am a student enrolled at San Francisco State University. So a lot of what my day-to-day looks like is because I'm trying to figure out the evolutionary relationships between some spiders in a family called Dictinidae. I spend a lot of my day in the lab extracting DNA, sequencing DNA. I spend a lot of my time on the computer analyzing my data. So I have some abilities related to these activities. But then if you go to the next slide, that you can see that I evolve also into a field form. In order to do all the studying that I'm so fortunate to get to do, I also go out in the field to collect the animals that then I take their DNA and I can ask some really interesting questions. So this is a little bit maybe a little less glamorous. I have some abilities like digging in the dirt and seeing at night, driving a long time, and also cooking on some camp stoves. So I'm going to tell you first about some spiders that I've been working on. Like I said, for the spider family Dictinidae, these are a group of little brown spiders that just not a lot of people know about what their relationships are to one another, and so I'm basically using their DNA to figure out how they evolved and are related to one another. So I want to introduce you to just one of the spiders that I'm studying. So if you go to the next slide, we have a little video that I'll probably narrate because I'm not sure what the sound is like. But this is called Argyronema aquatica, also known as the diving bell spider. And you can see it crawling, and it's called a diving bell spider because it spends a lot of its time actually underwater, and it makes an air bubble so that it can still breathe like any other spider. So it spends a lot of its lifetime in this, and it's just, yeah, it's really cool just to see how it's behaving and living its life. One of the other cool things about it is that it can take its prey. It can capture its prey and then bring it underwater and then bring it back into that air bubble and then use its venom to immobilize it. And it's much more potent in the air sac rather than in like the water where it would get diluted. So yeah, that's the diving bell spider, just one of the really cool spiders that I get to work with. Some of the other spiders are in some of the hottest places in the world. In Death Valley, they sustain some of the hottest ground and air temperatures. So those are different ones than these. But others live in like Greenland, and they're in the snowy tundra. So just within this family, there's an incredible amount of diversity, and we just don't know a lot about how these spiders are related to each other or how these really diverse adaptations arose because we don't have that phylogeny. All right, yeah, you can see some of the prey capture going on inside the bubble. Great. Okay, next slide, please. Awesome. So if you're familiar with Pokemon, you may be familiar with Dewpider, which was inspired by this spider and also lives in an air bubble as well. So in order to study
not this particular species, but other species in the family, I have had to do some field work. So if you go to the next slide, I will show you some pictures from my very first field expedition. So about a year ago, in my first year of my master's, I got to travel to the southeastern U.S. And visit, about 7 sites all along these states. We started in Atlanta and we drove to New Orleans over the course of 9 days, stopping in various forests and parks and things to just look for spiders that are in this family that I'm trying to study. And I've never been to the southeastern U.S., so it was really cool to see the landscapes there and meet some folks out there and really get to know more of the United States. All right, next slide. So when I was there, I was schooled in the ways of catching all the spiders. So there's a couple different tools of the trade that we use. One of the ways you can look for spiders is you can just go out into a forest and just look with your eyes and see what you can find under a log or on a tree or whatever, and just pick them up with your hands and put them in a vial. You can also use a tool called a pewter or an aspirator, which you can see in my mouth. So this actually uses suction to suck up the little spiders or other small invertebrates that you might be looking for. Don't worry, there is a screen so they don't go in your mouth actually, but you just suck them up, and then what we would do is then spit them back out essentially into a tube of ethanol or alcohol. So this does kill the spider, but we do end up sacrificing them so that we can take their DNA later anyway. So we just do it right away. Another way that you can use the pewter is you can use it in conjunction with this thing called a beat sheet, which is this white, it's essentially just like a white piece of fabric that is pulled taut over some PVC pipes, and you hit a tree as vigorously as you can, and then stuff from the tree or the bush will just fall on this white sheet of paper, and then you can look and you can see what's there, and then it's easier to then suck up what you need to suck up. We have also, you can brush a tree with a broom. These are all things that you can just buy at a hardware store. And then you can look and see what's there. You can also collect leaf litter, because a lot of spiders will actually live on the ground in leaves. And then you can take that back and use another device, which I'll show you in a couple slides, to get the spiders out of that. So these are a few of the tools that we use. There's also my colleague Franklin. He's showing me some of the spiders that he found as well. And there's a really cool illustration I just found on Instagram of what spider collecting looks like as well. All right, next slide. So sometimes when you're beating, you actually also catch things that you don't mean to catch. In this case, I was beating a tree and a frog fell in my beat sheet, so that was really exciting for me because I also like frogs. We also caught a black widow, which was unexpected, not really the kind of spider that we were looking for, but just also cool to see. All right, next slide. So then when you're done with your day, you've been out in the forest in Georgia all day, you go back to the Best Western and you set up your lab in the hotel room. And this is kind of what it looks like. We have, use the desk that they have there, and you have your little field microscope, and you're gonna start looking at what are the spiders that we have today. You need to look at them closely in order to be able to identify them, because many of these spiders, especially the ones that I'm interested in, are only sometimes 3 millimeters big, so they're really tiny, and you definitely need that microscope. Another thing that we do is we take the leaf litter and we put them in these devices. They're sometimes called winkler traps, but basically how they work is that there's a screen in the middle of this fabric hanging device, and you can put the dirt and the leaf litter on top of it, and then you hang an incandescent light bulb, not one of the energy smart ones, the one that actually get hot, and then the heat causes the spiders that spend most of their time in the leaf litter, in the dirt, in the cold, wet earth to migrate away from that. But then at the bottom, you have a tube of ethanol waiting, so they walk to their own demise. But this is how we actually filter a lot of the leaf litter specimens as well. So that's kind of a snapshot of how we catch some of these spiders. If you go to the next slide, please. One of the other great things about fieldwork is that you get to eat a lot of cool food. So like I said, we were in the southeastern U.S. and I got to try a bunch of great barbecue and stuff. And Franklin, my colleague there, was really into it, so I had to show food. And then finally, the next slide. So the final step of collecting is that then we take it back here to the museum at the California Academy of Sciences. And I spent a long time going through all of the specimens that we had gotten and putting them in the vials that correspond to their family. So somebody else who has a little more expertise than me, like actually sorted them and said, oh, this is a Honiad, this is a Ferridiad, this is like all in these different spider families. But then I spent many hours putting these little spiders into separate vials, and then that's how we organize them, and we put them in the collection for ourselves to use, and for anybody else who works at this institution or another museum or university, or just anybody who has an interest and wants to study these arachnids in some way, they can come and look at these, and they're all organized in a way that's actually helpful for them to use. And yeah, we ended up with 450 vials, so I listened to a lot of audiobook hours, but it was great. All right, so next, I'll just quickly also talk about another project that I've gotten to work on. This was a project that was largely led by Jacob Gorneau, who just spoke before me. And he was doing a project where he's trying to figure out how scorpions, their density and how well they've been doing has been affected by wildfires in California. So there's a data set that existed of scorpion sampling before some of the big wildfires that have occurred, and then we went in and tried to find as many scorpions as we could post-wildfire to see if there's a difference. So Jacob ended up going to over 100 locations, and I got to join him on some of those locations, and we went and looked for scorpions. So next slide. So one of the cool things about scorpions is that they actually glow in the dark if you have a UV light. So if you have a UV flashlight and you shine them on them, they will glow green. And this actually makes it really easy to find them at night, because if they're just crawling around and you have a UV flashlight, they just pop out, and you can just take your big forceps or your big tweezers and pick them up, or you could pick them up in your hand if you prefer. These ones that we're looking at, Euoctonus mordax, the western forest scorpion, they're not particularly venomous. It would hurt if they stung you, but it's you're not in trouble. So if you go to the next slide, I'll show you just kind of some of the fun and not so fun parts of fieldwork. Also, you get to see these obviously like really cool animals. There's obviously some cool food, some weird food that you end up hodgepodging together from various grocery stores in the middle of nowhere. We were out running some wildfires at the time, trying to get away from some wildfires that were cropping up. We actually hit a bear with our car at one point. The bear was okay. We made sure of that, but it was kind of a harrowing experience. Jacob fell in a hole once. There's lots of adventures and misadventures of fieldwork, and here's a few illustrations of that. All right, next, please. And then just finally, I want to tell you about one more project that involves some field work for me. So this is a project that I did in my undergrad. I made an observation of this frog and this newt in amplexus, it's called. This is the typical pre-mating posture that the frogs will undergo. So I think this frog got a little confused and thought that it could mate with this newt. But it turns out they are not in the same egg group, so they cannot breed. But what I was interested in finding out was that these newts are pretty poisonous. They secrete a toxin called tetrodotoxin. So I was worried for this frog that it had been exposed to some toxin. So I wanted to find out if there was some genetic evidence that potentially these frogs had some resistance to the toxin. So in order to do that, next slide please.
I surveyed a few ponds in the East Bay. This is in Brownies Regional Park, and I went along here and just took surveys of how many frogs and how many newts I saw in order to see if their proportions were such that I would think that the frogs would be exposed to enough toxins to have resistance. Next slide. Here's just a couple other photos from my field excursions. My lovely assistant Katie is in the audience today. This was a couple years ago. She came and helped me out, look for some frogs and newts. And in the end, we didn't find any evidence of resistance in these frogs to the toxins. So either they're not actually exposed to the toxin as much as we think they are, or there's some other mechanism resistance that we can't see just by, we did some genetic work on them and we couldn't find any evidence, but we tried. All right, next slide. So in sum, you've gotten to meet some of the really cool creatures that I have had the pleasure of catching and looking for in the wild. And next slide. But you can too on iNaturalist. So this is a really cool app. I'm happy to tell you more about it after the talk too. You can go on your phone and download it. It's like you take pictures of any organisms that you see and anywhere in the wild, in your backyard, wherever, and it actually has artificial intelligence that can help you identify those organisms. And then when you upload that observation, it really does help scientists. They can use that data. I've used data from iNaturalist in my own work, and we just have a better understanding of what organisms are occurring in our own backyards. So I really encourage you to get outside and look for the Pokemon in your own backyard using iNaturalist. So happy to take any questions if we have time. Thank you.
Kate. Thank you so much, Kate. We do have a little bit of time. I do wish you could see these Twitch comments, though. When you showed the salamander and the frog and amplexus, someone said, "Love wins." Someone from our Twitch audience wants to know, what does the diving bell spider eat if it lives underwater? Like, what underwater prey does it catch?
Yeah, that's a great question. So because I work with so many spiders in this one family, I don't know all of the details of all of their lives, but I think that these ones actually will catch prey that doesn't necessarily live underwater, but they can catch it and then bring it back to their underwater livelihoods. But I think I would imagine they could also catch underwater invertebrates as well.
Do we have any audience questions? Cool.
I'm wondering if you know what eats the diving bell spider underwater, and if you showered before or after putting the traps up in the hotel.
Thank you. The first question is, I don't actually also know what eats the diving bell spider. I would imagine it would be frogs and maybe some fish. And I would take my shower before putting up the traps because they would go all night long. We would leave them on. Yes.
And lastly, people want to know what audiobook recs you have.
Right now I'm listening to Viola Davis's autobiography, and it is excellent. So that's what I recommend. Thank you.
Thank you so much, Kate, for your talk. If you're just joining us live here at the Academy, or if you're joining us live on Twitch, we are hearing lightning talks that are all Pokemon themed from California Academy of Sciences scientists and researchers. And we're also at
Hi. Actually, I'm just gonna... Hello, how is everybody doing? I hope you're all excited to learn about reptiles and amphibians of the Pokemon world. Oh, there we go. All right, so we're gonna be playing a little bit of a game. Who's that Herp Pokemon? I will tell you now some of the silhouettes are misleading. Next slide. I'm just going to do a brief introduction. So my name is Chelsea Connor. I'm from the Commonwealth of Dominica. It's this small island right here in the middle of the Lesser Antilles. It is not the Dominican Republic, which is that way, completely different country. I am a graduate student here at Cal Academy, and I study reptiles and amphibians. I have done a focus on unknown lizards, which we'll talk a little bit more about as we get into this. Next slide. So, this was supposed to be an animation, and it was supposed to ask, who's that Pokemon? Clearly it is not playing. Next slide. So it's supposed to be Trico. It's Trico. Next slide. And Trico is a gecko Pokemon. It is based on a leaf-tailed gecko. We actually have one in the rainforest. It's probably closed now, but you can come in anytime and check it out. And they're called leaf-tailed geckos because of their tails. They are actually shaped like leaves, and their bodies do resemble drying or dead leaves. They also have these like really cool appendages on their jaws and on their fingers and toes. That helps them really blend in. If you've ever seen our leaf-tailed gecko here in the museum, it just looks like if it sat on the bark, which it does occasionally, it will blend in really well, and they do. It's really hard to spot them once they are actually sitting on a tree, which is why I've picked these pictures to show you, or you wouldn't have seen them. Trico itself, as a Pokemon, does spend a lot of time in the trees, and it does hide and blend in really well there. Shape of its tail also resembles a leaf, which helps it hide from, any potential enemies or trainers trying to catch it. Next slide. Again, this animation is not going to work, so we're just going to go next slide. It's Snivy. Snivy and its evolution superior are based off of lizards. You might think that superior here is based off a snake. No, it's actually based off a legless lizard, and that's what these two pictures are. The first one is Britain's legless lizard. It's one of many species of legless lizards. Over time, there are some lizards who have just lost their legs in the same way that snakes have. So snakes are technically lizards as well. But these ones, you can tell, they just look really weird. You'd be like, oh, that's a snake because it doesn't have legs, but then you look at it closer, and you're like, there's no way that that is actually a proper snake because it looks too strange to be one, and you would be correct. Looking at the one on the bottom, you can see that its body just looks too weird. It looks out of place. It should not be here. And actually, you would be right, because its body is really short, and the whole long bit that you see is its tail. That's one of the major differences between snakes and legless lizards. They have a short body and a long tail. So if you were to dissect it, you would see that all of its organs take up a short amount of this whole winding body. If you were to dissect a snake, then its organs are going to be through the full length of its body, and its tail is going to be about this short. For this legless lizard, I'm going to walk over real quick. For this legless lizard, its body probably ends right about here with its line ends, and this all is tail. And you can imagine how it would look with legs. The tail would make sense then. And the one at the top I included because I really like the shape of its face. It's one of the reasons why we think that legless lizards lost their legs, because it's easier to burrow when you don't have legs. You can just shove your face into the soft dirt or leaf litter and then just keep going. Plus this one looks kind of goofy. I just like that. All right, next slide. Yeah, next slide. So next up is Salandit. It's actually one of my favorite Pokemon ever. Next slide, please. So Salandit is actually based off of a salamander. I think this might be the fire-bellied salamander over here, which is toxic. So if you handle this salamander, you have to wash your hands before you put them anywhere near your face or mouth because the poison could harm you. Salandit is a fire and poison type. That's where that comes from. The name Fire-bellied Newt is because of the red coloring on its stomach. But then on the picture on the bottom, that is also a salamander in Old English mythology, which is where the fire typing comes from. And that belief comes from the fact that they would cut firewood and then toss it into the fire, and the poor salamanders that were hiding in there for safety would come out. The heat would like drive them out, and people would think like, oh, this must have been born of the fire. If you play like old Japanese RPGs, you will also see that old English mythology in there, in that like the fire element animal that you get is like a salamander. It's a dragon named Salamander, and that's where that comes from. Next slide. Yeah, next slide. Another one that I think is like actually a really good representation of the real animal is Turtonator. And it is based on the Mata turtle. It looks really goofy. I really enjoy that. I love a good goofy looking animal. And as you can see from the picture on the top, it's full body, especially when it's in the water, like it usually is. It resembles like a piece of old bark. And then it has some appendages that, unfortunately this picture is not moving, but if it were, you would see them like floating and moving around in the water. So it sits there and it looks like an old piece of bark, and fish swim by. And when they get close enough, it opens its mouth really quickly and it contracts its pharynx so that it sucks water in, so it sucks the fish in, and then it closes its mouth really quickly and swallows the fish whole. Because of how its derpy face looks, it does not have teeth, so it just has to swallow the entire fish whole, sadly. But as you can see from the actual Pokemon and from the animal, they did a really good job of representing it. I feel like they could have made a water and ground type instead of fire and dragon, but that's just me personally. I think it still looks great. Next slide. Next Pokemon is Heliolisk, and it's actually based off of like more than one lizard, it and its pre-evolution. So the first one is the frilled lizard, and the one at the top here is the frilled lizard. It's doing a display. It's trying to ward off like enemies or predators by trying to make itself look as big as possible. Heliolisk can also do that with this frill around its neck, so it will like open that up and try to look as big as possible, as threatening as possible. We call that a threat display. On the bottom is the Basilisk lizard. If you have played a recent Pokemon game like Pokémon Snap, Heliolisk can actually learn surf or run underwater, and that is based on this Basilisk lizard here. So the common Basilisk is also called the Jesus lizard because it can walk on water. And how it does that is that it has like webbed toes. So it will move really quickly and it will slap 1 foot down onto the surface of the water. And because of that webbing, it will catch air and push the air bubbles down. And the air bubble will help keep it buoyant. And before it can lose that buoyancy, it will slap the other foot down and it will just keep going. So it's really just like faith and luck and air bubbles that keeps it like going as it runs across the water. It will go slower or sink faster if it is an older or larger basilisk. So the young ones are the ones that are best at doing this. Next slide. Oh, sorry, quick mention of the last lizard, Heliolisk. It also gets power from the sun. And that is what a lot of lizards do. That's why you see them basking in the sun. They need that vitamin D and the sunlight to help them digest their food and help them raise their metabolism and be able to get energy from the prey that they consume. So next is Cyclozar, and it is one of my new favorites. It is also the middle form of the past and present legendaries that you get in Pokemon Scarlet and Pokemon Violet. So if you have Pokemon Scarlet like I do, you'll have this guy right here, which is Koraidon, and then Miraidon is the future version of it. It's a really great view of evolution, and if you want to learn more about that, I'm going to do a quick shameless plug. I co-host a Pokemon podcast called PokéScience, and we talk about this Pokemon specifically and how this is a really good reflection of real life evolution. But Cyclozar and its two other forms are based off of lizards that have dewlaps, anoles. So the two top photos are anoles. The one on the left is a chameleon anole. They're called that because they mimic chameleons. They're not actually chameleons. And then the one on the right is a Caribbean anole, and it's actually flashing its dewlap. It's that flap of skin under its neck. And they have a like specialized hyoid bone that they can contract and retract that will like allow them to display this, and they display it to attract females and to ward off other males, and to communicate to like predators or scientists trying to catch them for research that you probably shouldn't mess with me. There's also a really cool photo of a green anole right over here that you can see, and they always look very sassy. That's one of the reasons why I like them. I did my undergraduate research on this group of anoles, specifically one species that lives on my island, and they are just really fascinating. There's over 420 recognized species of them, and out of those, only 2 of them have no dewlap, but they use other ways to communicate. There are other lizards that have dewlaps as well. The Savada and Satana lizards, those are the ones on the bottom. They are found in India, and they have really large dewlaps. But as a personal note, I think anoles are much better at doing the dewlaps. In the Pokemon Cyclozar, you can see that dewlap communicated as like the wheel of the Cyclozar, in addition to like them having another wheel that is their tail when they curl it up. And I just think it's a really, really good representation for the first time of a lizard I've loved forever. I've been waiting so long for Pokemon to like introduce an anole, and it finally did. And it's also like legendary. It's like, this is great. I love it. I'm having a great time. You might have a question about this one eating a snail. The chameleon anoles also eat snails. They have specialized molars that help them crush the shells of those. So they're also called snail-eating anoles, and people who keep them as pets have to like keep buying snails for them to keep eating because it's their favorite thing to eat. All right, next slide. All right, that's it. Thank you so much, and you can look for PokéScience wherever podcasts are podcasted. Thank you.
Thank you so much, Chelsea. We have some great questions from our audience on Twitch, but if you're here in person, you have any questions, just raise your hand. We'll try to get to as many as we can. Someone on Twitch, they mentioned that like only female salandits evolve into salazzle. Is there any like real-world sexual dimorphism in salamanders that might have inspired that?
So I'm not sure about salamanders, but there are whiptail lizards that are only female. They have no males. They just clone themselves and have babies. So they lay eggs, and those eggs are all female. They do not need males. Sometimes they will stimulate mating, but they don't actually need to. They can just like produce their fertilized eggs and lay those eggs, and they hatch into more females.
That's absolutely wild.
Lizards are great. I love them.
Yeah, obviously. Someone else on Twitch asked if you could make any herp into a Pokemon that hasn't already been made into a Pokemon, which herp and what type would it be?
Oh, that's a great question. Okay, hold on. I really need to think about this one. I think I'm gonna have to go with it's another leaf-tailed gecko, but it's a satanic leaf-tailed gecko, and it's like a dark red and brown, and it has like eye spikes. It just looks really like evil, like an evil version of Trico. Think about that. And I really want that because I loved Trico so much when it came out. I just like would like an evil version of it.
My question is, if you need to, how do you catch an anole?
So it's kind of like fishing, but on land. Literally, I have a fishing pole, and I have, yeah, and I have like fishing line attached to it. It's tied in like a little lasso, like the tiniest lasso about this big, and I look for an unsuspecting anole, and I kind of like ease the lasso over its head. And then when I think it's in a good spot, I just have to yank it really quickly. It doesn't hurt the anole. They just kind of hang there bewildered, like, what just happened? Did somebody just pull me out of my tree, but there's nobody touching me? and then like I reel it in and take the lasso off, and then take any measurements that I need to of the pictures. Think about like you're just sitting here on this bench, and then all of a sudden you go flying across the room. That's what it's like.
The Twitch chat is blowing up about parthenogenesis. like, please tell us more.
I don't know what else there is to say other than, sometimes you need a man.
True. Do we have any more questions in person? No? Okay. Thank you so much, Chelsea, for the presentation.
Thank you.
All right. I think it's sad, but that was our last lightning talk of the night. We have to close it out. It's a bummer. I think, but thank you so much for joining us, whether you're in person or you joined us live on Twitch. We really appreciate it. Thank you for being so active in the chat. Yeah, thank you, and enjoy NightLife.